So in this lecture, we are going to discuss introductions of the amino acids. Amino acids are basically building blocks of the proteins, or you can say proteins are actually made up of different amino acids, and these amino acids actually link through the peptide bond, or you can say peptide linkage. So if you see here are basically the four amino acids. Here is the amino acid 1, here is the amino acid 2, amino acid 3 and amino acids 4, and these amino acid link through the peptide linkage, or you can say peptide bond. So here is the peptide first peptide bond. Okay, here is the second peptide bond, and here is the third peptide bond. So if you notice that the number of peptide linkage will be one less than the number of the amino acids. Here are basically the number of amino acids are actually 4, but the number of peptide linkage is actually 3, one less than the number of the amino acid. If I say if you have 10 amino acids, then the peptide linkage will be 9. Okay, that's the most important thing. So if we discuss the structures of the amino acid, that is the most important thing. So at the center of the amino acid, you will see the presence of the carbon, and at the side chain you will see the presence of the carboxylic group. Okay, that is the most important group attached to the central carbon atom. Okay, and at other side you will see the presence of the amino group. Here is the amino group. Okay, and at the third side or third corner of the carbon you will see the presence of the hydrogen, and at the fourth side or fourth corner of the carbon you will see the presence of the R group, and R group will be different for the different amino acids. Okay, if I say if I replace this R with the hydrogen, then it will become the glycine amino acid. Okay, so it's mean that R group will be different for the different amino acids. Okay, so this central carbon is actually called the alpha carbon. This is the alpha carbon. So why do we call it alpha carbon? Because it directly attached to the functional group, carboxylate group, amino group. So that's why it is actually the alpha carbon. Okay, this is the normal structures of the amino acid, but at the physiologic pH or at the 7.4 pH, now at that pH you will see the some changes in the basic structure of the amino acid. So if you see, central carbon will remain the same. Okay, and at that side you will see the loss of one hydrogen. Now at the carboxylate group you will see the one hydrogen will remove, and now this hydrogen will go towards the amino group, and in that side when you see the loss of one hydrogen, then that carboxylic group will carry the negative charge. Okay, and the other structure will remain the same, hydrogen will remain the same, and the R group will remain the same in the amino acid. And at that side, at the amino side, you will see the NH2 will become the NH3 and the positive charge on the nitrogen. Now this is the composition at the pH, physiologic pH 7.4. You will see the transfer of the hydrogen atom or hydrogen ion from the carboxylic group to the amino group. Now this composition is actually called the Zwitter ion. Okay, that is actually the Zwitter ion. Okay, having equal number of the positive and the negative charge. Now this is the compositions of the amino acid at the 7.4 pH. Okay, so it will act as acid and base. When you add acid to the Zwitter ion, then it will act as base, and if you add the base, then it will act as acid. So if you see here is the structure of the Zwitter ion. Carboxylate group will carry the negative charge and the amino group will carry the positive charge. if we add the acid, and acid actually produce the H positive. Okay, now if you see here acid, then that will be the H positive. Now that H positive will bind to the carboxylate group. Okay, now you see the attachment of the, here is the carbon, here is the C double bond negative, here is the hydrogen, amino group, NH3 positive, here is the R group. Now the hydrogen will attach to the carboxylate side, and in that situation you will see only the presence of the positive charge. Okay, and that will happen in the acidic condition. It means that in that case you will see the pH will be less than 7. At 7 you will see the neutral, below the 7 you will see the acidic pH, and above the 7 you will see the basic. Okay, next case, if you add the base to the Zwitter ion, then what will happen? Okay, now in that case you will see it means that you are adding OH negative, because OH negative is actually provided by the base. Okay, now in that case you will see here is the carbon, here is the R group, here is the hydrogen, and here is the carboxylate group. Okay, now in that case you will see one hydrogen will release from the amino group, that will be the H positive. Now this H positive will attach to the OH negative and result in the formations of the water. Okay, here is the formations of the water, and now at that side you will see the amino NH3 will become the NH2. That's the most important thing. That will happen in the basic condition, that will happen in the acidic condition. Now in that situation the pH will be greater than 7. Okay, so in the acidic condition you will see the presence of the positive charge on the amino group, and in the basic condition you will see the presence of the negative charge in the carboxylate group. But don't look at this, these are basically neutral, positive and the negative. Okay, if we see how different amino acid link through the peptide bond, so here is the amino acid 1, here is the amino acid 2. Now if we open the carboxylate group COOH, then it can be written as this one, COOH. And if we open the amino group, then it can be written as NH2. Okay, the other structure will remain the same. Now in that situation you will see the OH group from the carboxylate group and the hydrogen atom from the amino group will react between them, and ultimately you will see the release of the water molecule. Okay, that will water molecule H2O. Now in that case you will see water molecule will release, and ultimately you will see the formations of the peptide bond between the carboxylate group and the amino group. Now this is the C double bond O, here is the peptide linkage, or you can say peptide bond, and here is the NH2. Now this will happen again in the left side, and this will happen again in the right side, and you will see the formations of lot of amino acid will combine through the peptide linkage. So this is all about the amino acid. If you still have any question, you may ask in the comment section. Thank you so much.